When you consume refined carbohydrates, within just 30 minutes, your blood sugar spikes dramatically. Contrary to popular belief, this doesn't satisfy hunger. Instead, the rapid glucose surge triggers your pancreas to release insulin. Insulin's job is to lower blood sugar by storing glucose as glycogen or converting it to fat. However, an insulin flood often overshoots, causing a rebound hypoglycemia. Your blood sugar plummets below baseline. This sudden drop sends distress signals to your brain, leading to increased hunger. The vicious cycle continues, as this hunger often drives you to eat more refined carbs, perpetuating the cycle of constant hunger. This is not mere willpower failure, but a biochemical feedback loop. High fructose corn syrup is pervasive, found in countless processed foods. Within 30 minutes of ingestion, your liver starts converting it into fat. Unlike glucose, fructose bypasses the insulin response, entering the liver directly. Here, it undergoes rapid phosphorylation, a chemical transformation that prepares it for conversion into triglycerides. This fat production doesn't just add inches to your waistline, it fails to trigger satiety hormones like leptin. Leptin normally signals fullness, but without its release, you remain hungry. This stealthy metabolic hack illustrates why high fructose corn syrup contributes to persistent hunger despite calorie consumption. Your body's satiety signals are effectively muted. Protein satisfies hunger more effectively than carbohydrates or fats. Within 30 minutes, amino acids begin circulating in your bloodstream. These building blocks of protein stimulate the release of peptide YY, a hormone that enhances feelings of fullness. Researchers found in a 2011 study published in the American Journal of Clinical Nutrition that meals with 30% protein increase peptide YY levels significantly. Yet, many people's diets are skewed towards carbs, leaving them less satiated. As peptide YY levels rise, your stomach relaxes, signaling the brain to cease the search for food. This mechanism shows why protein is an underrated ally against constant hunger. Artificial sweeteners might seem like a calorie-free solution, but they trick your brain. Within moments of tasting sweetness, your brain anticipates calories. This expectation sets off insulin release despite no sugar intake. Insulin, being the diligent regulator, lowers blood sugar, but without actual glucose, you're left craving more food. A 2013 study in the journal Appetite found participants using artificial sweeteners ate more later. This mismatch between sweetness and caloric content leaves your brain unsatisfied, fueling further hunger. The perceived zero-calorie benefit is undermined by your body's expectation and subsequent demand for real sustenance. Fat delays gastric emptying, keeping you fuller longer, yet it's often vilified. Within 30 minutes, dietary fats slow down stomach contractions. This delay gives your body time to absorb nutrients and release cholecystokinin, a hormone signaling satiety. A 2010 study in the Journal of Nutrition found that meals with fat increase cholecystokinin more than low-fat meals. Paradoxically, low-fat foods often lead to overeating because they lack this hormonal response. Your stomach's delayed emptying is a crucial mechanism for maintaining satiety. This explains why fat, in controlled amounts, can be an effective component of a diet designed to curb hunger. Inside your stomach, ghrelin, the hunger hormone, rises before meals and drops after eating. But in 2014, researchers in cell metabolism found that consuming high-sugar foods disrupts this cycle. Sugar intake can suppress ghrelin's post-meal decline, leading to persistent hunger. As sugar enters your bloodstream, it doesn't just sweeten your palate, it alters ghrelin secretion. This alteration reduces your natural satiety signals, prolonging hunger long past your last bite. The mechanism shows how sugar hijacks your hormonal feedback loops, leaving you feeling empty even after eating. Ghrelin's mismanagement is a key player in continuous cravings. The gut microbiome, a vast ecosystem inside you, plays a surprising role in hunger regulation. Bacteroidetes and firmicutes, two dominant bacterial phyla, influence energy extraction from food. A 2018 study in Nature found that a higher firmicutes ratio correlates with increased calorie absorption. These microbes break down complex carbohydrates, enhancing caloric yield. If your gut harbors more firmicutes, even identical meals yield more energy, misleading your hunger signals. This microbial efficiency can warp your body's perception of fullness, contributing to overeating. The unseen world of bacterial interactions within you is a hidden driver of hunger management. Entering the bloodstream, leptin, a hormone produced by fat cells, communicates with your brain, but high-fat diets can desensitize leptin receptors. In 2012, researchers in Science Translational Medicine showed that excess saturated fat blunts leptin's signal. As your brain receives less leptin input, it perceives an energy deficit, triggering hunger. The cascade of events begins with excess lipid molecules infiltrating brain tissue, disrupting receptor function. Your brain, confused by the skewed signal, increases appetite despite adequate fat stores. This leptin resistance reveals the intricate hormonal ballet disrupted by dietary choices. In your intestines, glucagon-like peptide-1, GLP-1, emerges as a critical hunger modulator. GLP-1 slows gastric emptying and promotes insulin release. A 2015 study in diabetes care highlighted its role in reducing appetite. Yet, diets high in refined sugars and low in fiber diminish GLP-1 secretion. Fiber-rich foods, on the other hand, amplify GLP-1 production, promoting satiety. This peptide's journey through your bloodstream ends at your brain, where it signals fullness. Understanding GLP-1's delicate balance illustrates why fiber-rich diets can effectively curb hunger in a way processed foods cannot. Within your brain, the reward circuitry involving dopamine plays into hunger. Sugary foods trigger dopamine release, reinforcing eating behavior. In 2017, a study in neuroscience found dopamine spikes resemble those from addictive drugs. These surges create pleasure pathways, encouraging repeated consumption. However, continual dopamine stimulation can lead to receptor downregulation, dulling the reward response. As a result, you crave more to achieve the same satisfaction. This neurochemical rewiring, driven by sugar, showcases how hunger can be influenced by pleasure rather than need. Dopamine's intricate dance illustrates hunger's complex psychological dimensions. Chronic overconsumption of calories can lead to a paradoxical state where your hunger persists. A 2020 study in the Journal of Clinical Endocrinology and Metabolism found that obese individuals often have elevated ghrelin levels, contrary to expectations. This persistent elevation means hunger remains even with excess body fat. Inside you, adipose tissue grows, but your brain misinterprets signals, maintaining the hunger cycle. Ghrelin's unnaturally high levels seem to stem from leptin resistance, where the brain ignores satiety messages. This metabolic confusion shows how prolonged caloric surplus reprograms your body's hunger signaling, perpetuating an endless loop of eating. Nutrient deficiencies, surprisingly, can also leave you feeling hungry. Magnesium, for example, plays a role in regulating insulin and blood sugar. A 2011 study in the Journal of Nutrition found that low magnesium levels correlate with increased insulin resistance. Without adequate magnesium, your cells struggle to metabolize glucose efficiently. This inefficiency sends repeated hunger signals to your brain as it seeks more energy. Despite calorie intake, the lack of essential nutrients like magnesium keeps your body in a perpetual search for sustenance. This nutrient-driven hunger showcases how micronutrient balance is crucial in maintaining satiety. Consuming excessive caffeine can also impact hunger, though not in the way you might think. Initially, caffeine suppresses appetite by increasing adrenaline. However, a 2016 study in the American Journal of Clinical Nutrition noted that habitual caffeine consumption leads to tolerance. As tolerance develops, caffeine's appetite-suppressing effects diminish. Inside you, the adrenal glands adjust their output, and hunger returns with a vengeance once caffeine levels drop. This rebound hunger can drive increased calorie consumption, often in the form of sugary cravings. The temporary nature of caffeine's effects on hunger highlights the importance of hormonal adaptation. Fast food companies have long understood how to exploit your hunger signals. Through strategic combinations of fat, sugar, and salt, they create hyperpalatable foods that override satiety cues. A 2013 study in the journal Obesity Reviews found these combinations stimulate the brain's reward pathways more intensely than whole foods. This stimulation leads to a form of sensory-specific satiety, where you crave more despite being physically full. Your body, overwhelmed by intense flavors, fails to recognize fullness. By designing foods that exploit these mechanisms, companies ensure you remain hungry for more, despite caloric intake. Finally, consider this. A 2015 study in Cell found that the brain's hypothalamus can actually remodel its neural connections in response to diet. This remodeling occurs in just a few weeks of high-fat, high-sugar intake. The brain physically changes, altering hunger signaling and increasing food intake. It's not just about willpower or metabolism, but a literal rewiring of your brain's hunger pathways. This revelation reframes the entire understanding of chronic hunger. The extent to which diet can reshape your brain highlights the profound, often underestimated impact of food on your biology. Your brain is not a passive player, it adapts actively to dietary patterns.